Hello students, good morning. My name is Rahul Gude. You can call me Rahul sir. And today we are going to see one topic from 12th standard that is called as Lac-operon. Let's try to understand what this Lac-operon is. First of all, what is an operon? Operon basically is a model. Lac-operon is also a model. Operon basically is a model of one of the thing like how it operates. If it is related with any particular gene, for example, here we are talking about lac gene, in that case, how this lac works, that particular model itself is called as Lac-operon. Jacob and Monod, these were the scientists who actually for the first time had given something related with the Lac-operon, how this Lac-operon works. What is Lac-operon? What is lac? What this particular gene is? This gene is something called as lac gene, and this gene basically works for degradation of the lactose. Uh, Before going ahead, let's talk something about a bacteria. Lac gene, it is present in the bacteria, especially in E. coli, Escherichia coli, in that this gene is present. Now the question is how this bacteria it is deriving its energy. For example, it is utilizing glucose from the surrounding, and from the degradation of that glucose, whatever energy that is evolved, this bacteria is utilizing it for itself. But when there is no glucose available, what will this bacteria do? Let's take for an example. When this bacteria is present in milk there is no glucose, but surely, certainly there is lactose present in it. Okay. Now what this lactose is? Lactose is basically made of two different components. First component is called as galactose, but second and most important content is called as glucose. When this bacteria bit is present in the milk, in that case lactose is available. Will it be better for this bacteria because there is no glucose, but lactose is there? Why not to extract glucose from the lactose? That's why with this bacteria, what will it do? It will try to degrade this lactose. Once this lactose it is degraded to glucose and galactose, in that case, this glucose it can be used and utilized for generation of the energy and for carrying out the cellular activities. Getting this point? That's why this lactose should be degraded so that glucose can be extracted out of it for the degradation of this lactose. For the degradation of this lactose into glucose and galactose, this particular gene it works. Lac is the word that has come from the lactose. Okay, so what is a lac-operon? It is the gene lac-operon. It is the gene present in bacteria which is used for degradation of lactose. Okay, and why this degradation is important? It is just because that particular bacteria it needs to extract that glucose from it. Okay, so with this view, let's go ahead. We'll first of all see how this particular gene is present. Now, as you all know that genes they are all located in the genetic material, especially in all organisms. The genetic material is called as the DNA, except viruses, some of the retroviruses in which RNA is a genetic material. But if we see the basic structure of this particular lac gene, in that case, there are some regions. You may be able to see that I am drawing something like P, then O, then I, then something Z, then Y, and then A. This is how they are arranged. These different regions they are arranged on DNA. Now let's see what this P stands for. P is actually stands for a region called as promoter. I region it represents here. I region it represents something called as repressor molecule. We'll talk in a while what is called as a repressor. If we talk about OO, it is called as operator region. Okay, and some other regions are also there. Let's see the Z region here. It the Z by A. All these three regions, they represent some enzymes. These enzymes, they are used for the degradation of the lactose. Z, it represent Z region here. It represent an enzyme called as beta galactosidase. Y region here on this DNA, this represents permease, and A region, it represents trans acetylase. Okay, beta galactosidase, it basically works for degradation of lactose. This actually cleaves or cuts the joining bond between glucose and galactose. It cuts that particular bond, and that is a glucose and galactose. They both are prepared. Permease, it is actually used for entry of lactose inside the cell. Okay, permease, it works for the entry of that lactose inside the cell. We'll talk in a while how this whole process is. And last one is called as transacetylase. Till today, transacetylase, the function, it is not clearly understood. But somehow it must be working in that. The function is actually clearly not known. It basically transacetylases. These are the enzymes which actually works for transfer of acetyl group. That's why they are considered to be transacetylases. But what is their application in the degradation of the lactose? It is still not known. So basically, these are different regions which are present on this particular strand of the DNA, and this particular strand of the DNA, it represents the Lac-operon or the model. This particular complete gene is called as Lac gene. Now let's see how this gene basically works. Okay. Now, as you all know, if you are aware, then you must be knowing something about protein synthesis. Protein synthesis, it is divided into two different parts. The first part is called as copying of mRNA from this DNA. Okay, copying of mRNA from the DNA is called as transcription. This particular process is called as transcription. Okay, so likewise, before going for transcription, let's talk something about promoter region. The basic function of the promoter region is not especially for copying out from the DNA to mRNA, but basically this promoter region, it works for promoting the transcription. Like for example, this promoter region, it is there. It attracts the RNA polymerase towards it. What is the function of RNA polymerase? RNA polymerase comes and gets attached on the DNA. From that particular DNA, it will be able to produce mRNA from this. Okay. Promoter region is never never transcripted. It is never converted to mRNA. It promotes the transcription. Getting my point? That means, in other words, you can call it as this is the attachment site for RNA polymerase. Okay, so whenever any RNA polymerase will come and get attached on this promoter region, it will start producing mRNA from the next region. Likewise here, if you see here, I region is there, which represents the repressor molecule. Then from this I region, there is production of this mRNA. From that production of the mRNA, there is a production of repressor molecule. Okay, this is called as repressor molecule. This, from mRNA to the production of this repressor molecule, it is called as, this process is called as translation. Transcription and translation, they both work. They both are the part of protein synthesis. Okay. So this I region, it has produced this mRNA. From this particular mRNA, this repressor molecule is produced. Correct? This much is fine. Now promoter region is also situated here, right? As the promoter region, here it attracted RNA polymerases. Likewise, this promoter region, it will also attract RNA polymerase towards it, right? Likewise, it will move ahead to from the operator region, and next there is three different enzymes. They can be produced by this one, correct? Now, if RNA polymerase will run from this particular RNA, what will be happening? In that case, it will be able to produce mRNA from this region, mRNA from this region, mRNA from this region. Likewise, from every mRNA, every protein will be produced. That means from this Z region, the mRNA is produced. From that mRNA, beta galactosidase is produced. From this Y, mRNA is produced, that is how permease will be produced, and from this A, mRNA is produced, and likewise transacetylases it will be produced. Getting my point? Now, the basic question is, can this production of these enzymes be continuous? If you remember at the very beginning of this lecture, we were talking about the degradation of the lactose. When degradation of lactose will work, when there is ample amount of lactose but there is no glucose. If glucose is available, why will the cell waste its energy? Why will that bacteria waste its energy? In that case, what will it do? It will simply stop the production of these enzymes because there is no lactose available. Complete glucose it is available. At that time, this gene is now no use. Getting my point? Now here is the crux. We can compare this thing with something else. Like you are having one company, maybe a biscuit company. You are able to produce biscuits more and more. Every day your company is producing 1,000 units, but there is no demand for the biscuits in the market. For especially for your biscuits, there is a demand for the biscuits, but not for your biscuit. Every day you are producing 1,000 units. Will you keep producing these units? Will you keep continuously producing these? Of course, you will not do that. What's the reason? Because if you will keep producing that one, nobody is nobody is there to purchase that. In that case, the production of these many number of biscuits, it will be of no use. It will be a waste of time, waste of energy, and waste of money. That's why you will stop it. Likewise, when there is no lactose available, in that case, these genes, from these genes, those three enzymes, they should not be produced. Why? Because when there is no lactose available, then these enzymes, where will they work? There is none of any substrate that is available for them. That's why production of these three enzymes it should be stopped. For that, repressor molecule it works. How does it happen? Very simple. This repressor molecule, it goes and gets attached at operator region. Now, speciality of this repressor is whenever it is made, it goes and gets attached at the operator region. If it is present at the operator region, what is the function of promoter region? It attracts RNA polymerases. Whenever RNA polymerase will come, in that case, it will try to run over this operator region. But when it will try to run over this operator region, there is the presence of the repressor. That's why this RNA polymerase, it cannot go ahead. Are you getting my point? Let me tell you again. What is the function of promoter region? To attract RNA polymerase. This repressor molecule, it is already produced here and it is getting attached at the operator region. Even if at this promoter region there is any RNA polymerase coming and getting attached, in that case, when it will try to go from this operator region, in that case there is a repressor molecule which is stopping this. That's why RNA polymerase cannot go ahead, and that is it will get detached. Now my question is, if RNA polymerase cannot go ahead, when RNA polymerase it cannot go ahead, can it prepare any mRNA from these three genes? It cannot. Because there is no mRNA, will there be any production of these proteins? Of course not. Getting? That's why, that's why attachment of repressor at operator region, this is simply called as switch off mechanism. Okay, attachment of the repressor at operator region, it is simply called as switch off mechanism. Okay, now this is the condition when there is no lactose available. Only during that this gene will be switched off. Now take for an example the E. coli, the bacteria in which this gene is present. This bacteria, it fell into a bowl containing complete milk. What will happen? Is there any glucose available? No. But is there lactose available? Yes. Now the bacteria will think that why not to degrade this lactose so that this extraction of glucose can take place, and from this glucose I can derive my energy. In that case, what should be done? So far as this operon is concerned, in this case, you will have to switch on this gene so that there will be the production of these enzymes, and so that lactose can be degraded very easily. Are you getting my point? Why this gene is switched off? It is because there is no lactose present, but ample amount of glucose it is present. That's why there is no need to degrade the lactose. That's why this gene is switched off. But when now there is presence of lactose, in that case you need to degrade that lactose. That bacteria needs to degrade that lactose. How will it be done? How can it be done? Think for a second. This repressor molecule, when it is getting attached at operator region, this is called a switch off mechanism. What will be switch on? Switch on means this repressor, when it is not binding at operator region, in that case, that will be called a switch off. Maybe you can say like you are removing this repressor from here, or in other words, not allowing this repressor to get attached at operator. Correct? Can that be called a switch on mechanism? Of course, it is called a switch on mechanism. How this basically works? It is very simple. Before some time, when we were talking about Z, Y, and A, at that time, I was telling you that three different enzymes are working. First enzyme was beta galactosidase, which actually cuts this lactose into glucose and galactose. But the second enzyme called as permease, what does it work for? It works for entry of lactose inside of the cell. Bacteria is present, outside is bacteria, complete lactose is there. This lactose, it needs to be transported inside of the cell. For that permease will work. Correct? But in a longer term, like later on. But before that, you need at least that bacteria, it needs at least few lactose molecules so that this process should be started in a better way. What will happen? Now, if there is any lactose molecule which is outside, the cell will try to take two or three molecules of the lactose inside of the cell. And what will be the function of these lactose? Now, RNA polymerase has come here. No second guess we are talking. RNA polymerase has come. It went to the I region. That is how it has produced one mRNA. From that mRNA, repressor molecule it is produced. What is the function of this repressor molecule? Its basic nature is to go and get attached at operator region. Now, before its attachment, if there is any lactose molecule, for example, this is called as lactose. If this lactose molecule gets attached with this repressor, what will happen? This is a repressor molecule and this is a lactose molecule. Okay, this is repressor and this is lactose molecule. Now what will happen? What was the basic function of this repressor? This repressor's basic function is to get attached at this operator region. Did it get attached? No, it got attached with lactose. Getting my point? Now tell me, if this repressor, it is attached with lactose, what is there? Can it bind at operator region? No. When it is not attaching at the operator region, what will happen? RNA polymerase, it has come on this promoter region. Now it is trying to go from this operator region. Is there any obstacle? Is there any repressor molecule present at operator region? No. When there is no repressor attached at operator region, what will happen? RNA polymerase will run over this DNA, and that is how it will translate this Z into mRNA, this Y into mRNA, this A into mRNA. And that is how from this beta galactosidase it is produced From this, permease is produced, and from this, trans-acetylase will be produced. Are you understanding? It's a very simple process. When this repressor it is attached at operator region, these three enzymes cannot be prepared. That's why attachment of this repressor at operator region is called as switch off mechanism. Now, if this repressor it is getting bind with lactose, in that case this repressor cannot get attached at operator region. So when removal of this repressor from the operator or not allowing this repressor to attach at operator region, this is simply called as switch on mechanism. So the switch is on. So attachment of repressor with lactose molecule, it is called as switch on mechanism. Okay, so lactose, it is what this lactose is basically doing. Lactose is not allowing this repressor to get attached here, and that is how gene is switched on. What this lactose is doing, do you know? It is inducing the production of these three enzymes. That's why this lactose it is considered to be inducer molecule. Getting this point? This is how this Lac-operon actually works in two different ways. One is called a switch off mechanism. How does it? How does the switch is kept off? Very simple. From this region, this repressor it is produced. This repressor gets attached as this operator region. This is simply called a switch off mechanism. If this repressor which is continuously produced and every repressor it is getting attached with the lactose molecule, in that case this repressor it cannot get attached at the operator region. That is how when it is not getting attached, these three enzymes they are produced. In that case it is simply called as switch on mechanism. Okay, this is how lac gene it works for the degradation of lactose for getting glucose from that for deriving the energy. This particular gene is called as lag gene. This is how it works. Thank you.